Hi, I'm Lizzie. I'm from CRUK RadNet, City of London, and today I'm joined with Professor Roger Greenberg from University of Pennsylvania. And yeah, are you ready for some questions?
I'm ready. Yes.
Okay, perfect. So for our upcoming seminar, you've chosen to focus on the communication between DNA damage and immune response. So I was just wondering if you could tell me a little bit more about why you've chosen this topic and why you think that it's an important subject to be talking about.
All right. I think it's a fascinating topic, and it actually had intrigued me for a number of years uh, without us making much progress in the area. And that changed, I think, when we took a systematic approach to it. So just for a little bit of a backdrop, it had long been known that DNA damage elicited inflammatory responses within cells that could attract immune cells to a site of damage. And it also had long been known that exposures to DNA damaging agents in humans or mutations in genes involved in the DNA damage response as well could result in inflammatory responses and even autoimmune disease. And so this was an enigmatic area that we didn't understand the molecular basis for. It seemed to occur with vastly different kinetics than the typical DNA damage response occurs. For example, when cells are irradiated and they now have double-strand breaks after that, they mount a response that occurs really within seconds, and it's over generally within hours. Whereas this immune response took days to really evolve and to a maximal level. And that's really was the start of our interest. There were other interests as well. My interaction with radiation oncologists at the University of Pennsylvania were also quite illuminating. And they taught me about how the response to radiotherapy in vivo in preclinical models really relied on an intact immune system. So if tumors were irradiated, um, they didn't shrink down very much in immune deficient hosts. And that really, I think opened my eyes to the idea that the response was quite in human beings or in any organism, multicellular organism, was quite a bit more complicated than many of the cell culture models that all of us use. And so that really is the backdrop to it. And in 2017, I think we had a breakthrough in this area to help us understand how this communication occurred.
There's quite a lot of interesting work that your lab's doing generally, and one of those things that particularly interested me was the work that you've been doing to develop new methodologies for visualizing the major steps of homologous recombination. Could you tell us a bit more about those?
Sure. Yeah, homologous recombination is, I think, one of the most fascinating processes in biology. It's so complex, and yet it emerged, in bacteria. Viruses use this to replicate their DNA as well. So it's an ancient process, and it's incredibly elegant how broken or damaged DNA can use an exact replica, a template on the sister chromatid generally to fix that DNA in a process that is, at least in principle, is error-free. So, my lab started out studying how BRCA1, the breast and ovarian cancer suppressor protein BRCA1, was attracted to DNA damage sites. And we were quite interested for a while in the, I would say, classical form of homologous recombination from BRCA1 and 2. But another aspect of homologous recombination occurs at telomeres in about 10 to 15% of cancers, and that is, I would describe it as a gain of function, non-canonical form of homologous recombination that is specifically allows telomere maintenance and cellular immortalization in a subset of cancers that are typically not treated very well. And that also was interesting to us. The breakthrough for my lab in this area was a former student, Will Cho, developed a system to synchronize this process so that we could create DNA double-strand breaks specifically at the telomere, and that initiated this alternative lengthening of telomeres non-canonical recombination process. Subsequent student in the lab, Robert Dilley, and a postdoc, Priyanka Verma, developed additional methods to, in real time, really not only see the mobility of these telomeres where they would come together and recombine, but also to quantify their products, which is new DNA synthesis or new telomere synthesis. That, really gave us, I would say, a general handle on the process. There's certainly much that we don't understand about it, but it allowed us, I think, to look at all of the basic steps as they were occurring, and we could take advantage of the repetitive nature of the telomeres to be able to see this very well. In unpublished studies that I hope to submit pretty soon, we've been able to purify the entire DNA damage response in the telomere and be able to test hypotheses from this DNA damage response proteome. And so step by step, my lab, as well as many others, are, I think, starting to put a dent into the molecular workings of how this alternative lengthening of telomeres process occurs.
Perfect. And how do you hope that your research will benefit patient outcomes?
Yeah, I think that's the overarching goal of many of our labs. What I try to do is keep it in perspective that we will pick problems that we think are quite important and where there are major gaps in knowledge and just follow scientific method. Um, what are the observations that can allow us to ask fundamental questions in these areas? How can we test these hypotheses and figure out how things can work? And if we can do that, then I think we're confident that we can actually have a translational impact on patient care. And I think that's a model that's worked very, very well in the DNA damage response for many, many labs. And for example, PARP inhibitors and much of the advances in radiotherapy and immunotherapy and others. It's an exciting time for basic science to be translated pretty rapidly. So, some of the examples from my laboratory, we have found that biallelic mutations in BRCA1 were the causative genetic event in in a developmental and hereditary bone marrow failure disorder called Fanconi anemia. That was one of the subsets of that. We've recently discovered a new target, ALC1, along with several other labs in the UK and elsewhere for BRCA mutant cancers. So that loss of this gene, ALC1, results in extreme hypersensitivity of BRCA mutant cells to PARP inhibitors at very low doses. And I would say our work with communication between the DNA damage and immune responses has also, I think, been influential in the design of clinical trials that are ongoing now to harness the immune response against tumors. And so, for example, we've shown that entry into mitosis is really the key gatekeeper to this communication, and that if you disrupt the G2 checkpoint, for example, with ATR inhibitors, this can result in a very robust response. On the other hand, if cells don't progress through mitosis, it really eliminates the response. And so there are ongoing clinical trials testing these combinations with immune checkpoint blockade, either PARP inhibitors plus immune checkpoint blockade, or radiation plus immune checkpoint blockade, ATR inhibitors plus radiation in ICB. So, empirically, this will be determined in the clinic. I view it as my mission to provide basic information that can be developed by others, either companies or by more translationally oriented investigators.
That's really interesting. And I guess more generally speaking, what kind of research developments do you hope to see within your field in the next, say, decade?
Those are often hard to predict, and it's frequently the unexpected finding that becomes so influential to everyone. But what I would say is there has been a tremendous wealth of information from model organisms to understand the DNA damage response. Work from bacteria, from yeast, from viruses, and with all of the developments in mammalian approaches in mammalian cells, at a cellular and molecular level, we're really starting to gain a good understanding of the DNA damage response at incredible detail. And that will continue in exciting new ways. Some of them, I think, predictable, some of them not. And there will be new concepts brought forward, like phase transitions and many other things. So that is one aspect that certainly will develop. But another really untapped area is, how does this DNA damage response signal at an organismal level, right? Human genetics tells us that mutations in a number of different DNA repair pathways can create vastly different phenotypes in humans, different genetic syndromes, bone marrow failure, cancer predisposition, neurodegeneration, right? Almost every aspect of human biology and human disease is touched upon by, germline mutations in DNA damage response genes. So how that works on this three-dimensional level and sort of at a tissue level and well beyond that in a system, I think is very ripe for discovery, and for discovery at the interface of different disciplines like immunology or stem cell biology, cell-cell contact. All of that are areas that are very, it's been, I would say, difficult to study, but I'm sure there's a wealth of information out there that we have not tapped into yet. So.
Perfect. And next week on the 24th of March, you'll be joining us and the rest of CRUK RadNet City of London for a seminar. And I was just kind of wondering what it was that you were most excited for your upcoming seminar with us.
All right. Well, I think giving seminars to these sort of audiences with broad expertise is always interesting. It forces one to think about different aspects and different angles to the work, different perspectives. And there's also the give and take with the audience, hopefully during question and answer, that might help change the way one thinks about a problem. Science is interactive. It has to be exchange of ideas. That's how we learn. We don't do it in isolation. And with all the things going on with the pandemic, one silver lining in the science has been that there has been a lot of this virtual communication. I think I've attended more seminars than I have ever had ever done previously. I've learned a heck of a lot that way, and it's nice to be able to contribute and receive feedback on my own work, and that's really what I'm looking forward to.
Oh, that's great. Well, we're looking forward to it as well. So thank you for joining us. I think that's all my questions today. You briefly touched upon it on your last answer, but if anybody else has any questions for Professor Roger Greenberg, then there will be a question and answer session in our seminar on the 24th of March. So to find details and to reserve your place, check out our website, have a look at our social media, and that's it for us today. So thank you for joining us again, Roger. It was an absolute pleasure, and see you next week, I guess.
Thank you. I'll see you next week, and stay well.